Hi. So we will continue today and we will talk about hemoglobin. And basically, we're going to focus on chapter 6, as usual, in our textbook, The Clinical Laboratory Hematology, third edition by Mackenzie and Williams. And today we're going to talk about hemoglobin. And our learning objectives are going to be simple. We're going to diagram the quaternary structure of a molecule of the hemoglobin and identify the heme ring, the globin chains, and the iron. So let's start. So first of all, what is hemoglobin? It's the most specialized intracellular red blood cell protein, and it's the protein that transports oxygen from the lungs to the tissue, but also facilitates the CO2 transfer from the tissue to the lungs. And it occupies 33% of the volume of the red blood cells, and it accounts for 90% of the red blood cells' dry weight. So each cell will contain between 28 to 34 picograms of hemoglobin. And we can measure that by cell analyzers that we will learn about in our next classes and even demonstrate using them. And the measurement is reported as the mean corpuscular hemoglobin, or what we call MCH. So the most hemoglobin is synthesized in the polychromatophilic normoblast, and we learned about the stages of development of the red blood cells. And to remind you that the polychromatophilic normoblast is this cell in which there is still nucleus, but it's not as big, and the ratio between cytoplasm to nucleus is increasing compared to the previous cells. But we still have the nucleus, so then it's 75 to 80% of the hemoglobin is made before the nucleus is extruded, and the rest of that, 20 to 25%, is made by the residual RNA and mitochondria in the reticulocytes. So to remind you about the stages of the red blood cell development and production, and I know that most of you answered the questions regarding the development in the quiz correctly, so we covered that subject really well. So basically, hemoglobin, we know from last week that red blood cells life cycle is 120 days. So basically, the hemoglobin concentration is a result of the fine balance between the rate of the red blood cell production and the rate of the red blood cell destruction. And to remind you, you guys can go after the class and watch this video on YouTube about the life cycle of erythrocytes. And you can see this balance between red blood cells production and destruction, and how important is that for functioning red blood cell system. So the normal concentration of hemoglobin in adults is around 15 gram per deciliter. And to remind you, the total blood volume is around 5 liters. So as I said, the normal lifespan of a red blood cell is around 120 days. So 1 divided by 120 is the total amount of hemoglobin that is lost daily. So we need to make around 6.25 grams of hemoglobin every single day, and to have 750 grams of hemoglobin in total. And we know that the mean cell hemoglobin, we said, is 28 to 34, so it's around 30 picograms. So we need to make around 2 multiplied by 10 to the 11 red blood cells every single day in order to maintain normal concentrations of hemoglobin in the adult body. So how is hemoglobin built? What is it? We said it's a protein and its molecular weight is 66,700 daltons, and it's composed of 4 globular protein subunits. And each subunit, we can see here 2 units are green and the other 2 other units are yellow. And in each subunit, we can find a heme that is a terapyrrole ring that the ferrous iron sits in the center, and it's depicted here as red sitting on a blue ring. So each chain of these subunits has a heme inside it. So we said that each heme subunit can carry 1 molecule of oxygen and is bound to the ferrous iron. So we can here see the ferrous that is making 6 connections, 1 with the oxygen and 4 with nitrogens and 1 with another nitrogen below. So each hemoglobin molecule can carry 4 molecules of water. We said that the hemoglobin is built of 4 structures of globulin chains, and there are 2 types of globulin chains that are produced, either alpha-like or non-alpha. So alpha-like can be either alpha or zeta, and these are made of 141 amino acid in each chain, whereas the non-alpha, which can be epsilon, beta, delta, or gamma, are a bit longer, and they are made composed of 146 amino acids in each chain. So the globulin chains create a tetrametric hemoglobin molecule that contains 2 pairs of unlike chains, and we have 2 identical alpha-like chains and 2 identical non-alpha-like chains. So in our example here, we have an alpha chain that is depicted in orange, another alpha chain that is depicted in purple, and 2 beta chains, one that is red or purple or rose versus one that is green. And each one of these chains has a heme molecule inside it. And the heme group is surrounded by the globulin chain and is held together by salt bonds, hydrophobic contacts, and hydrogen bonds. And they form a tetrahedral formation, meaning like this is a tetramolecular geometry where the heme is sitting in the middle and is connected by 4 poles. So the major that we can look at the hemoglobin structure according to the things we learned in molecular classes, according to the primary structure, the secondary structure, tertiary structure, or the quaternary structure. And when I say primary structure, to remind you, it's the sequence of the amino acids. And we said that the alpha chain is much shorter than the beta chain. The secondary structure is the one that creates either the alpha helix or the beta pleated sheets that are formed by hydrogen bonds between the peptide bonds in the chain. And these are here. You can see here a helix. Tertiary structure is the folding of the molecule into a 3D structure, and you can see it here, in which the heme sits in the middle, whereas the quaternary structure is the combination of all the 4 polypeptide subunits, 2 alpha and 2 beta, and which each one of them contains a heme group into a larger protein. So the big protein is made of 4 subunits. And here is a table that I added to the presentation for you to summarize what we talked about till now about regarding the structure of the hemoglobin. For next lesson, we will explore abnormalities in hemoglobin structure and how they affect how they have clinical manifestations. So in order to be prepared for next week's class, let's all take a few minutes to read this review paper and look at the hemoglobin variants and the biochemical properties and how they correlate to clinical findings. So I'll see you in 2 hours after we read this paper. Thank you so much.